Okay, we are now recording this second Zoom session on the inner ear anatomy. We're continuing from where we left off last week, and we covered there some of the gross anatomy of the inner ear. I will share screen here, and we'll get to take a peek at some of that. So let's see what we've got. Last week, we did talk about, first slide, we talked about the bony labyrinths, the membranous labyrinths, and we talked about the vestibular system here in pink, and about the cochlear system here in orange and yellow. We then highlighted the hair cell regions of each, really stressing the membranous labyrinths of each, the vestibular system and the cochlea. We said these are the utricle and saccule. We called these the semicircular canals. We went on to describe the membranous labyrinth of the cochlea, and then went into a bit of detail talking about two different types of hair cells inside your vestibular system, namely type 1 and type 2. We said that type 1 hair cells are afferent and type 2 hair cells are efferent. And in the cochlea, the corresponding hair cells would be called the inner hair cells, which are afferent, and the outer hair cells, which are efferent. We'll have more of close-ups of that in just a bit. Anyway, highlighting the vestibular system, we'll go to our notes here for a second on page 1, and we dispensed with the vestibular system fairly quickly. So we went on to describe that the sense of balance has 3 parts: vision, proprioception, and the vestibular system itself. We described each of those, and then we said, let's look at the vestibular system itself, and it is comprised of 3 parts: the utricle, saccule, and semicircular canals. We described what each of those are generally for. We described the hair cell regions inside the utricle and saccule, called the maculae. We described the ampullae, which is the membranous labyrinths areas of the semicircular canals. And and inside this maculae, inside the utricle and saccule, and the ampullae inside the semicircular canals, are hair cells. Those are the membranous labyrinths areas, the hair cell regions. Then we described 2 different acronyms in balance testing. One was electronystagmography, ENG, and benign paroxysmal vertigo, BPV. Now, if you look over here, you'll see those exactly. So these areas that I'm highlighting are things that I think people should have put a star by to make sure that you are able to gamely describe what each of those are in general. What does BPV stand for? What basically is it? What does ENG stand for? Who does it? Audiologists, not HISs. Okay, so then we dispensed with the vestibular system, and we talked about the gross areas of the cochlea, the basic anatomy of the cochlea. So, in so doing, keep going. Here we go. Membranous labyrinths, bony labyrinths, and I said this whole white area is solid bone, and this gray area is a tunnel carved into bone, and the membranous labyrinth is much like a hose snaked into the curved hole that I dug into the bone. Okay? If you're looking at the membranous labyrinths over here in the center, you can see where it ends. You can see where the utricle and saccule are, the bags or the squeezy areas. Here's the ampullae areas of the semicircular canals. So hair cells in here, hair cells in here, hair cells, hair cells, hair cells, hair cells, and hair cells inside the cochlea. Good. Membranous labyrinth, looking closer or more detailed there, you can see it's highlighting the membranous as opposed to the bony labyrinths, which is highlighted here. And then you can really see the specific hair cell regions inside of each of those. And then I said, let's look at a little bit more finely, and you can see that it's not really so much of a round hose, it's more like a triangulated shape. And I highlighted this very carefully because I said when you look at it closer up, you'll really see that triangular shape. And you'll really see it, and you can see that this membranous labyrinth here, this membranous labyrinth serves really to split the bony labyrinths in half. And you can really see that here. It splits the bony labyrinths in half. This is just a picture looking at the membranous labyrinths and how it communicates with the 8th nerve. Membranous labyrinths of the vestibular system and how they communicate to the 8th nerve, all of this going toward the brainstem. A cross-section of a cat's cochlea, and here you can see again bony labyrinth, and the membranous labyrinth splitting the bony labyrinths into two. So one coil, another coil, another coil. Basically this wrapping around to here, wrapping around to here, wrapping around, and so on, okay? This would be the cochlea unrolled. And so now you're taking this whole thing and you're simply unrolling it. And again, you can see bony labyrinth in white, helicotrema, which is the communications area, and then bony labyrinth here. Stapes pushing into the oval window, pushes fluid here around, which bulges the round window out. So if the round window can't bulge out, the oval window can't really push in. You need that interchange to occur. The promontory is really the bony ridge in the middle ear space that separates the oval from the round windows. So this is just a schematic. And notice how the membranous labyrinth gets wider as you get toward the apex and narrower as you get toward the base. So this area has more mass and less stiffness. This area has less mass and more stiffness. This area on the left therefore resonates with low frequencies. This area resonates with high frequencies. And in that way, the hearing aid, the cochlea could be said to be tonotopic. Specific frequencies are represented in specific places, much like keys on a piano. Okay, and we'll talk more about that in cochlear physiology when we go there in the next 2 weeks. This is a cross section of the cochlea. This is my way of drawing the cat's cochlea. Okay, but look at it from the cat cochlea to my drawing. Look at the similarities. The membranous labyrinth gets wider and wider as you go toward the narrow end of the cochlea. The membranous labyrinth gets wider and wider as you go to the apex of the cochlea. So it's exactly back-asswards from the way one would think. This is a point that we need to take home, okay? So now we can look at some of the gross anatomy of the cochlea, and let's look at this slide. This is actually out of a hearing aid manufacturer, okay? And I've taken away the name of the hearing aid manufacturer, but now as OTC students, you should be able to label what's wrong with this picture. And you can see the cochlea there, nicely coiled up, 2.5 coils, nothing wrong with that. You can see the foot plate of the stapes in the oval window. Here they call it the stirrup. Anyway, in the oval window, you can see the round window, nothing really wrong with that. But what is wrong with this picture is the fact that the scala media is drawn to get smaller, or actually that it doesn't widen as you get toward the apex of the cochlea. That's the flaw in that picture. But they are just showing for the lay public how this stapes pushes fluid around and around and around, and it goes through the helicotrema, la 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 la, down, out, out, out to bulge the round window. So in that wise, there's not a whole lot wrong with the picture, but technically there is. Here's another picture. It's a cross section, and they're doing the same mistake here. They're showing the scala media actually larger here and getting smaller as you're going up, which is just exactly the opposite. Now, what's good about this picture, though, is it highlights that bony core inside the cochlea. So that bony core is called the modiolus, M-O-D-I-O-L-U-S, modiolus. It's a core of bone, but look at what's inside of it. You can see these round bulges, and these round bulges are the 8th nerve leaving the cochlea. And if you look toward the bottom, you can see the 8th nerve leaving the cochlea, but look at the cross section. Because you can see a bulge here, one here, one here. Now, what the heck is that? If you look at that carefully, you'll see it in my drawing too. Modiolus is the bony core inside the middle of the cochlea. And again, you see this ball, another one, another one. Now, what's with those? That's what I want to highlight here. Well, that's this. See, what you're looking at here is the 8th nerve as it leaves the hair cells, and it goes out toward the modiolus, which is over on the left here, and you've got one arm of the 8th nerve, which is where my cursor is, and then you've got these little olives, and these look like little pimentos. They're looking at you, okay? Well, those are the cell bodies of the 8th nerve. And when you think of the 8th nerve, I'll just draw a picture of it here. If you draw a picture of any neuron, and I'm just going to draw this up for you and then hold it up so you can kind of see what I'm showing here, okay? Whenever you're drawing a picture of neurons, you can see one arm, and you see a cell body, and you see the other arm. So this arm is talking to something, this arm is talking to something else. Maybe this arm is connected toward your brain or spinal cord, and maybe this arm is going to your legs or to your arms, okay? So it tells you you can pull your muscles. And the cell body is a nucleus, and the cell body, that's what feeds the neuron. And the neuron is a fiber. And nerves are bundles of these neurons, like thousands of them, like a rope. Okay, when you think of a rope having tons and tons of fibers, well, that's the 8th nerve. And that's why they show the 8th nerve in pictures, often looking like this. Okay, it's like it's a bundle of neurons leaving the cochlea. Huge bundles of them. See, look at this. Here's thousands and thousands of those fibers. And they're leaving the inner hair cells. Okay, the inner hair cells are afferent, and we'll show more of that as we move along. So here's one coil of the cochlea. We're just taking one of these guys and we're pulling it closer up. Or actually, to be more honest, we're taking this one. Okay, pulling it closer up. Here's the scala media, membranous labyrinth. This is Reissner's membrane, basilar membrane, and I said last week, this especially, the basilar membrane is the floor upon which the hair cells stand. Okay, it's the floor upon which the hair cells are all standing up there. Okay, so here's inner hair cells, 3 rows of outer hair cells. These are supporting cells. And these are supporting cells. And this is the tectorial membrane. It's a gelatinous mass that sits right on top of the hairs of the hair cells. And this is the stria vascularis we said last week. It's the blood supply and the oxygen supply to the hair cells. Reissner's membrane, you could bend it. Blah, blah, blah. It's the membranous labyrinth. You can squeeze it, okay? And this is filled with endolymph fluid. This is filled with perilymph fluid. Perilymph fluid. So the basilar membrane is held tight as a drum, between this spiral ligament on the right and spiral lamina on the left. And we call them spiral because they continue all the way up the cochlea. That's why we call them spiral. And here's the cell bodies of the eighth nerve, the thing I showed you earlier on the picture here. Okay, the cell bodies are these. One arm is going to the hair cells. The other arm would go up toward the brain stem. So why is it that pictures are showing you 1, 2, 3, 4, and 5? Well, that's because this is a cross section. In all reality, this whole situation is going, so you got a bulge here, a bulge here, a bulge all around, all the way around, thousands of them. So basically taking a cross section, you might see 4 or 5 just because it's one cross section. Again, I'm showing you in my picture 1, 2, 3, 4, 5, 6. But again, if you could look 3D, you'd see these all just going all the way around. And that is why they are called the spiral ganglion. A gang of cell bodies. Okay? And you've got bulge, 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 bulge. Those who indulge bulge. You've got these bulges of them going spiraling all the way up the cochlea. And you have the spiral ligament going all the way up the cochlea. And the spiral lamina, the shelf of bone. And this shelf of bone going all the way up the cochlea. So 3 spirals. Spiral ligament, spiral lamina, spiral ganglia. Something decent to remember. Here's a closer up of it. And again, now they're showing you the 8th nerve a little bit closer. And you can start to see that spiral ganglia. Now you really get the feeling you're being watched. Here's all your little olives with the pimentos looking at you. Here's the inner hair cells. And the inner hair cells are mostly in cahoots with the 8th nerve. Now, the 8th nerve is a sensory nerve. Remember we said before, nerves can be either afferent or efferent. Sensory information goes from the body to the brain. Okay, that's what the word afferent. A-F-F-E-R-N-T means brain-going information. And brain-going information is vision, taste, touch, smell, hearing. All your sensory information goes brainward. What goes out? Muscular information. So they call motor cells or motor neurons efferent. They're going, their messages are from my brain or spinal cord to my limbs. So sensory in, motor out. Good. Inner hair cells are sensory. Outer hair cells are motor. Inner hair cells talk to the 8th nerve. Outer hair cells don't really. They don't communicate much with the 8th nerve. Now let's draw this picture in closer. Look where my cursor is. See these little neurons? They're going to your outer hair cells, but they're not really part of your 8th nerve. They're part of another nerve that runs along the 8th nerve. The 8th nerve is the big interstate highway. I don't know the one that goes from St. Louis, Missouri to Kansas City. I can't remember the name of that highway anymore, but it's about 4 or 5 lanes. It's a big super state highway. Well, think of this. That's the 8th nerve. Now think of a little gravel country road running alongside the interstate. That's the nerve that communicates to the outer hair cells. And it goes from the brain stem to the outer hair cells, and that's this. The inner hair cells send all info to the brain. The outer hair cells take info from the brain. So anyone who's been to Deutschland before knows now that the cochlea is nicht eine Einbahnstraße, okay? It is not a one-way street. It is a two-way street. You've got brain going and brain sent. Everything is like that, okay, in the body. So outer hair cells are efferent, inner hair cells are afferent. Now let's get closer. Inner hair cells, outer hair cells. Now this is an old picture too. There's a mistake in this picture. And it's no one's fault, it's just what people used to think. A more modern picture is that, no, that's not it either. Okay, a more modern picture would be this. Inner hair cells are fat couch potatoes. Outer hair cells are long and lean. Outer hair cells look like test tubes. Inner hair cells look like pears. Now, there's a second thing. Outer hair cells, the hairs, touch the underside of this tectorial membrane. Inner hair cells, the hairs don't. Okay? And we will learn more about this in physiology in the next couple of weeks. There's a reason why, but I'll just tell you now and a little bit ahead of time. Outer hair cells die before inner hair cells. Outer hair cells help inner hair cells pick up sounds below 50 decibels. If the sound's below 50, the inners can't pick it up. So the outers shrink, pull down, and they pull this tectorial membrane downward so these hairs can get bent. If the sounds are more than 50, there's sufficient fluid motion in the cochlea to wiggle those hairs by themselves. But if the sounds are less than 50, the motoric action of the outer hair cells is required. Somehow the brain knows and tells them to shrink. Since they're the moving part, they die first. And when they die, you have an inability to hear sounds softer than 50. And what hearing loss is that? Presbycusis. Presbyterian, Church of the Elders. Presbycusis, hearing of the elders. Okay? Presbycusis, trouble hearing treble. They can't hear high pitches. What's the degree of loss? 50 decibels. Where's the damage? Outer hair cells. Why? Because they die before inners. Why? Because they're the moving part. We'll discuss this more in good old physiology. Told you the ear is kind of weird, man. All right, so here's the inner hair cells. Here's what's wrong with this picture. The hairs are touching. Okay, that's what's wrong with the otherwise, and the hairs, inner hair cells should be fat. And the outer hair cells should be drawn long and lean. You can date audiologists like rings on a tree. If they graduated before 1990, they think all hair cells look the same. If they graduated after 1990, they learned the difference between the outer and the inner hair cells. I graduated in 1988 with a master's degree. I didn't really learn the difference between them. It was only when I went to the University of Oklahoma in the 90s that I learned what the difference was, because that's when the difference was found. Anyway, look closely here and you can see tectorial membrane, supporting cells, outer hair cells, inner hair cells. Now, this is at the base of the cochlea, because if you go to the apex of the cochlea, the hair cell region gets wider. Okay, where are the hair cells located in this picture? Right here, right here. Right here, okay? And notice that that region gets wider as you go toward the apex. So at the base of the cochlea, you have one row of inner hair cells, 3 rows of outers. In the middle of the cochlea, you've got 1 row of inner hair cells and 4 rows of outer hair cells. At the apex of the cochlea, you have 1 row of inner hair cells and 5 rows of outer hair cells. Just FYI, thought I'd tell you. Okay, so here you go, closer in, and you can see now the neuron fibers connecting to the outer hair cells. You can see the 8th nerve fibers connecting to the bottom of the inner hair cells. And look at the areas right here, right where I'm circling. Now this area is filled with fluid, and this area is filled with fluid. And this area is, but what kind of fluid do you think it is? It's not endolymph. Endolymph fills this area, okay? And endolymph fills, follow my cursor, endolymph touches the hairs of the hair cells and fills this area. But what's inside this white area here? Perilymph. Perilymph inside this membranous labyrinth is called cortilymph. Now, anyone who's been to Germany knows that years ago you had East Germany and West Germany. And East Germany was communist, but you had a capital city called Berlin. And in Berlin, there was an area called West Berlin. And West Berlin was capitalist, not communist. So it was like a little island inside of communist East Germany. You had to take a certain highway to get to it. It was highly guarded. So I always look at West Berlin like cortilymph inside the scala media. Okay, like it's an island of perilymph inside the scala media. So it's perilymph inside here, and it can't leak out. It cannot leak out. Look at this line. It's locked solid. Okay, so if you're looking at that close, it's locked solid. Think of hair cells as like my head stuck in a pillory. You ever look at the pilgrims in New England? What did they do to bad people? They put them in these things, right? They locked the wrists, and they had the head stuck in them, and people got to throw tomatoes at them. I can't remember what they called the pillory. They pilloried them. Well, the hair cells are pilloried. The necks are pinched off from the bodies. So the body of the hair cell is touching cortilymph. The body of the hair cell is touching cortilymph, and cortilymph is perilymph inside the scala media. They call it cortilymph, okay? And yet the hairs are touching endolymph. And the two cannot mix because it's pinched off. And that is how hair cells are in contact with opposite fluids, okay? Because you need that opposite fluids to create an electrical charge, because batteries have opposite fluids in them too. That's what creates a charge. And that's what the cochlea is supposed to do, change sound into electricity. And that's how it's done, okay? So if you're looking here, if you think, well, gosh, how can perilymph mix with endolymph here? Well, it can't, and it doesn't. So how do hair cells get that contact to perilymph? Well, they do it by means of touching it with their cell bodies inside the scala media is perilymph. And perilymph inside the scala media surrounding the bodies of the hair cells is called cortilymph. So the hairs are surrounded by endolymph, the bodies are surrounded by cortilymph. Just thought I'd lay it down. Now you're looking at the opposite. What we've done is we've taken away the hair cells, and you're focusing now on the supporting cells. Again, organ of Corti, but what we're doing now, we've taken the hair cells out. So this was the inner hair cells, one row of outer hair cells, another row, and another row, and we've taken them out. And this is just to show you how the hair cells are pilloried. So the cell itself is stuck in that round area and pinched off at the neck, and then just the hairs are into this area where my cursor is touching. Okay, so even in anatomy, all spaces are given names. Space of Newell, inner tunnel of Corti, inner sulcus. We got all this blah, 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 blah. Anyway, basilar membrane, spiral ligament, where my cursor is here, okay? Over to the right would have been spiral ligament and all that stuff. So spiral lamina, spiral ganglia, spiral ligament. Okay, here's another view of the organ of Corti, and all you're doing here is looking at it a little bit less flat, with a little bit more three-dimensionality. Okay, so once again, inner hair cells, you can see the shape, pear-shaped. Outer hair cells, longer and thinner. And now look at the hairs on the top. When you're looking at the hairs on the top, the inner hair cells, the hairs are all in rows. Whereas the outer hair cells, the hairs are arranged like little W's or V's. And that's an important thing to grasp as well. We'll look at that more. Now here's a picture of an actual human cochlea from someone who's died, okay, and you can see bony labyrinth, perilymph, Reisner's membrane, stria vascularis, the blood supply, spiral ligament, SL, okay, outer hair cells. These would be the inner hair cells. This is the tectorial membrane, but it has shrunk. It's pulled way back, but this would all be the organ of Corti. This would be the basilar membrane. And then this would be the spiral or the bony labyrinth, scala tympani, scala media, scala vestibuli. So let's look at that in color because it's way more fun. So here you are again. Bony labyrinth, Reisner's membrane. This contains perilymph, scala media, membranous labyrinth, contains endolymph. You've got your stria vascularis. You have your spiral ligament, this big fan-shaped thing here, basilar membrane, spiral lamina. And we can't see the spiral ganglia. That would be maybe more to the left. You can see one row of inner hair cells, shaped like pears, 3 rows of outer hair cells. These would be supporting cells. And here would this white thing would be the tectorial membrane laying over top. So this is a picture really close up now of the inner hair cells and 3 rows of outer hair cells. This must be near the base of the cochlea. So now you're looking at things from the top. Okay, so you're looking at things looking down. You've torn off this tectorial membrane. You ripped it apart. You took it off. And you're looking down at the top of the hair cells. Inner hair cells, you have 1 row of inner hair cells all the way up the spirals of the cochlea. You have 3 rows of outer hair cells at the wide base, 4 rows toward the middle coil, and 5 rows at the very top coil. And look at the way the hairs are arranged. Now this is damaged hair cells. This would be in someone who's done a lot of target practice or noise-induced hearing loss, or the person who has aged, presbycusis, and the damage is mostly to the outer hair cells. Outers tend to die before inners. See, the inners aren't so bad. It's mainly the outers that have the damage. Here's another picture of inner hair cells, and you're looking at 3 rows in this picture of outer hair cells. Here's another picture of inner hair cells, and you're looking at 3 rows of outer cells. On the right, you can really see the test tube shapes underneath. So you're looking from the top. See how they're pilloried? Okay, the hairs are kept separate from the cell body. The cell body is surrounded by cordolymph. The hairs are surrounded by endolymph. Now you're looking at the close-up of 1 inner hair cell. So you're looking at the cell from the top, okay, and then you can see the small choir boys in the front, the next taller ones, and the tall choir boys standing in the back. And that's how the hairs are arranged. And you can see how these are just not hairs, like hairs on a head. You can see how they end in a dull, rounded sort of edge. Here's the close-up of hairs of the outer hair cells. How many are there? About 100, 110. On the inner hair cells, about 50. Okay? How many inner hair cells do you have in a cochlea? 3,000. How many outer hair cells do you have in each cochlea? 12,000. Okay, it's not 9, it's not 3 times as many, because remember, you may be seeing lots of photos with 3 rows of outer hair cells, but remember, there's also 4 rows and there's also 5 rows of outer hair cells. So 12,000 outer hair cells in each cochlea, 3,000 inner hair cells in each cochlea. How many hairs on each inner hair cell? 50. How many hairs on each outer hair cell? About 100. Now here's a human hair. Here's a hair cell. And don't forget, on the hair cell are 100 hairs. And that's why I always say hair cells really are like the hair on the wart and the frog and the log and the hole in the bottom of the sea. They are small. Here's a picture of a bug under an electron microscope. Now the bug has in one of its claws a gear. Look closely at the teeth of that gear. Remember, this is under an electron microscope. So the bug is blown up, it's tiny, but it's got a tiny little gear in one of its claws. Bring it closer, and now you'll see a little white dot on one of the teeth of the gears. And that is a hair cell. Again, going back, back, forward, forward, and please don't think that's one hair. That's 100 hairs, but they've all died, and so they're all clumped together. Okay, we'd be talking small. Okay, so this is showing you a picture as to how the cochlea is tonotopic. Specific frequencies, if this is the basilar membrane, rolled out to be about 1 inch long, or north of the border, 2.5 centimeters in length, because it's about how long the cochlea is. The cochlea, if you unrolled it, is actually about 3 centimeters long. Okay, and you can see how high frequencies are located toward the base, low frequencies are located toward the apex. Now, let's go and look at our notes, because it's high time we made sure that what I'm blabbing about, we've all covered in our notes. So looky here. I think this would be the middle of page 2. Cochlea, Greek for snail shell, a winding organ with 2.5 turns. And then you'll see chinchilla cochlea is often shown. And we said last week the chinchilla cochlea is often shown because it has 3.5 turns and because it sticks up and into middle ear space. Hang on, I'm yapping too loud. My better half is going to hear me. I got to close the door here, so hang on a second. Oh, okay. All right. So, all right. Now, human cochlea, we said, is remarkably inaccessible. It's buried inside the hardest bone of the body, the petrous portion of the temporal bone, like an auger twisted hole. We've talked all about the scala tympani, scala media, scala vestibuli. We talked about the helicotrema. Then we showed you photos and PowerPoint slides of a close-up of the single turn of the cochlea. So make sure that you can identify each of these terms. Reissner's membrane, basilar membrane, spiral ligament, stria vascularis, modiolus, spiral lamina. Good. Move on down to page 3. Spiral ganglion. There's your third spiral. And then we went to a close-up of the scala media. And here we talked about tectorial membrane, gelatinous mass on top of the hair cells. We said how the tops of the outer hair cells are embedded in it, but not the inner hair cells. That's all in this section. Now, here's something you don't need to know. The area I'm highlighting right here. I couldn't care less if people knew about cells of Held, cells of Claudius, cells of Henson. I really don't care. Okay, I just want you to know that inside the organ of Corti there are supporting cells, and the supporting cells hold up the hair cells. Okay, but you do need to know cortilymph. There you go, inside cortilymph fluid inside the organ of Corti. All right, moving down page 3. You'll see the electron microscopy slides that I showed you. Here you go. This is the close-up of the hair cells. So this section is describing what we talked about in terms of hair cells. Inner hair cells and outer hair cells looking very different from each other. That's because they do vastly different things. More about this in physiology next time. Here's where it says the inner hair cells are shaped like jugs or like pears, right here. You have about 3,000 of them. They're all in one row, all the way up from the base to the apex. You have about 50 stereocilia on top of each inner hair cell. And we said the tips of the stereocilia are not, repeat, not embedded in the tectorial membrane. And then below that it says the outer hair cells are shaped like test tubes. You have 12,000 of them. in 3 rows at the base to 5 rows at the apex. And we said there's about 100 stereocilia on the top of each outer hair cell. And lastly, we said that the tips of the stereocilia are indeed embedded in the tectorial membrane. Now, what are stereocilia? The hairs. Stereo means many, cilia, hairs. And now look at this. Cochlea is tonotopic. And we've said that word before too. Specific frequencies are represented in specific places. High frequencies are located at the fat base of the cochlea near the oval window. Low frequencies are located near the apex of the cochlea. Now think about this. You've got the footplate of the stapes constantly activating the oval window, and you've got the cochlea spiraling up from here. What hair cells are going to receive the most action most of the time? High frequencies. Because everything's got to go through those highs to get to the lows that are at the apex. So the low frequency hair cells tend to be most protected. That's why highs die first as well. You can think of high frequency hair cells as like the carpet at the doorway where you rub your boots. Because that carpet by the doorway, everyone has to go through to get anywhere else in the room. Okay? And so people tend to get high frequency hearing loss before low frequency hearing loss. And that's why most presbycusis is trouble hearing treble. Good hearing in the lows, and then outer hair cell damage near the base of the cochlea. Okay, the cochlea is tono-friggin-topic. Never forget it. And the guy who said so, his name was Helmholtz. Helmholtz, place theory. In English, this just means specific frequencies are represented in specific places, just like keys on a piano. Same, same, same. Okay? Specific frequencies represented in specific places along the basilar membrane. Hair cells near the base of the cochlea represent highs. Hair cells near the apex of the cochlea represent lows. There's another theory, and it's called the temporal firing theory. And the temporal firing theory says, nah, the place theory isn't really correct. We have a sort of a different way of describing things, but it's the number of times that the eighth nerve fires per second. If it fires 100 times a second, you hear 100 hertz. If it fires 500 times a second, you hear 500 hertz. But, you know, we'll talk about that mañana. For main thing, it's the Helmholtz place theory. We're now winding down toward the end of our anatomy section on the cochlea. We're getting to the top of page 4. Here we're at the very bottom of page 3, and let's read this together. Eighth nerve is a bundle of sensory afferent nerve fibers. As such, it is mainly associated with the inner hair cells. Its cell bodies are located in the spiral ganglion of the cochlea. You've seen that now. Olivocochlear bundle, OCB, is that little country gravel road running alongside of the interstate between St. Louis and Kansas City. And that bundle, that neuron or that nerve runs along the eighth nerve, but in the opposite direction. And it goes from the brain to the outer hair cells. So the olivocochlear bundle, OCB, consists of efferent nerve fibers. They run along the eighth nerve, and as such, it is mainly associated with the outer hair cells. Efferent OCB messages course in the opposite direction to those of afferent eighth nerve. Okay, inner hair cells are sending info to the brain, outers are getting info from the brain. Inner hair cells are afferent, and guess, look at this, they comprise 20% of hair cell population, but they get 95% of the eighth nerve fibers. Each inner hair cell gets a bunch of eighth nerve fibers. To be specific, excuse me, to be specific, each inner hair cell gets 10 eighth nerve fibers. So if each inner hair cell gets 10 eighth nerve fibers, how many eighth nerve fibers do you have? Well, 3,000 inner hair cells times 10 is 30,000. So now you know that that rope called the eighth nerve has 30,000 tiny fibers wound up together. And now you look at the eighth nerve, and how does it communicate with the outer hair cells? Barely. A few fibers from the eighth nerve go to the outer hair cells. But you know what? One fiber from the eighth nerve is shared by about 10 outer hair cells, whereas each inner hair cell gets 10 eighth nerve fibers. So you can see that the eighth nerve is mainly in cahoots with the outer hair cells. And if I look at this in terms of a PowerPoint slide, I may have it. Oh, this picture is a nice one showing you that the cochlea is tonotopic. Okay, low frequency sound waves, apex of the cochlea, mid frequency, middle of the cochlea, high frequencies toward the base of the cochlea. Showing you again, high frequencies, lower and lower and lower as you get toward the apex. Here's a spiral ganglion, a real actual spiral ganglion. So this arm here would be going toward the inner hair cells. And here's the eighth nerve fibers, and here's the cell bodies looking at you. And this arm here would continue on toward the brain. This is a picture showing you inner versus outer hair cells and how spoiled the inner hair cells are. 10 eighth nerve fibers are connected to each inner hair cell. So you see that? One inner hair cell has got 10 fibers with 10 cell bodies sending info along these arrows to the brain. Meanwhile, one eighth nerve fiber is shared among 10 outer hair cells. So basically, the eighth nerve is an afferent system, essentially. It has a few fibers that talk to the outer hair cells, but not very many. Now, here's a picture. I may have shown you this in acoustics class. I can't remember. I get mixed up from classes to classes, but here's a guy who lives in a van down by the river who was a hunter. And he's got high-frequency hearing loss, second most common cause of hearing loss in the world. And the thick line is inner hair cells, and the 3 dotted lines represent the 3 rows of outer hair cells. So, look at what it says on the bottom. Apex on the left, base on the right. So this is the basilar membrane unrolled. And you can see that along the apex of the basilar membrane, no damage. But toward the base, where the high frequencies are, hearing loss. And you can see that all hair cells here are busted and damaged. If you look at this picture here, you can see a hunter who I love to say, he came to autopsy. I always love the way medicine says, he came to autopsy. What do you mean? Oh, he died. What do you mean? Here, he came to autopsy. Yeah, I need to die now. I always I think that feeling's hilarious. You see that puppy again. There he is. You got it made. You got 2 of them. 3 of them. Good stuff. All right. Gotta like that. Keep you out of trouble. We'll see who takes who out for a walk. It ain't you. All right. Somebody's got to be the boss. So now you're looking at a right-handed hunter, and this person here, when someone is shooting a long rifle, okay, a right-handed person, the left ear will be closest to the sound, and so this guy's right ear has a bit of death to the inner hair cells, but mostly to the outers. Whereas the, the left ear's got no damage to any hair cells until you get to the high frequency region, and then they're all dead. So it's just showing you a picture that way. So anyway, we have now reached the end of our anatomy notes, the end of anatomy of the cochlea. We spent 2 weeks on the anatomy of the cochlea. Next week and the week after, we'll figure this out. I don't know when your midterm, you should be getting a midterm coming up. The midterm will probably cover outer ear, middle ear, and a bit of the inner ear. Some, I would probably say up to the anatomy of the middle ear, of the inner ear. It won't, I don't think it covers the physiology of the inner ear. That's a whole different kettle of fish, and that too takes a couple of weeks to go through. So we spend 4 weeks on the inner ear. You've done 2. So I'll talk to Ms. Kraus, to Becca Kraus, and see what she's doing regarding the midterm exam. In the meantime, I encourage any of you in the class to inquire to her as well as to what is generally covered on the midterm. But to the best of my knowledge, it's outer, middle ear, and inner ear anatomy only, not inner ear physiology. So, this is, you are now in the thick of it. Where do we go after inner ear anatomy or physiology is done? We start talking 8th nerve and brain. And then things get a little bit easier. Actually, those areas are a little bit easier because we know less about them. The tough stuff really is the anatomy of the inner ear and the physiology of the inner ear. That's the most detailed stuff. All right. Bark in German for us here. Gesundheit. Good stuff. All right, I will cut it short here. If you have any questions, don't hesitate to ask me. And if you don't, you don't. But if you do, you do. If you leave me an email, I check my OTC emails every ding-dong day, so I'll get back at you as soon as I can. Every time I see myself in this thing, I always see this one eyebrow stuck way up. I look like Spock on Star Trek or something. You know what that was? When I was 6 years old, I fell off a toboggan and cut my face. They did stitches, but they didn't do the stitches very well, so my eyebrow sticks up all the time. That was a need to know. I knew that you needed to know that in order to pass the course, so I thought I'd share that with you. All right, big events in the lives of little men. I will now stop recording and end the session, and we'll talk at you when we do next, okay? Ciao for now. Live long and prosper.